Hello, everyone. We're ready to get started. Welcome. I'm Annie Razovitz, the Communications Advisor for the F3 Future of Fish Feed team. I'd like to introduce Dr. Kevin Fitzsimmons, Chair of the F3 team, who will be moderating today's webinar. As many of you may know, Kevin is a professor at the University of Arizona, the former president of the World Aquaculture Society, and currently leading an EU project on sustainable aquaculture in Myanmar. I'll hand it over to you, Kevin.
Thank you, Annie. We want to welcome everyone and thank you for joining us today for our single-cell novel proteins and aquafeeds. This is the third in a series of monthly F3 webinars we're hosting for the latest advances in aquaculture feed ingredients to replace fish meal and fish oil. I think you all realize this, but we need these replacements. Wild-caught fish are a declining resource, and without replacements, aquaculture is bottleneck and cannot grow to feed a world of 9 billion people. But replacements are around the corner. Our seminars feature the most promising ingredients so that aquaculture can continue to grow sustainably. For today's session, we have major players from the single-cell and novel protein sector. There are many unique and exciting new technologies, in part accelerated by the search for COVID vaccines, that are being deployed to deliver tailor-made solutions to both the human and animal nutrition sectors. These techniques are enabling nutritionists worldwide to create customizable nutrients that are more efficient and economical and reduce feed and production costs for fish farmers. We're excited to learn about some of the top innovators and producers in this space today. So let's get started. First, we have Dr. Larry Feinberg, who's the CEO and co-founder of NIP Bio. Larry is an entrepreneurial industrial biotechnologist with years of experience in biotech industry. Some of his ventures include leading the Organism Discovery Group and the New Business Opportunities teams for Mascoma Corporation, which is an advanced biofuel technology firm. NIP Bio is a blue biotech company developing responsible and sustainable commercial feed solutions for the aquaculture industry. In 2021, NIP Bio is launching its first of a kind FDA-approved product to market. We look forward to hearing more about this innovative solution. So, Larry, let's hear from you, please.
Hi, I'm Larry Feinberg. I'm the founder of NIP Bio. Delighted to tell you a little bit about the company, introduce myself, and be part of the Future of Feed meeting here today. So a little bit about NIP Bio very quickly. We are a blue biotech firm based out of Boston, Massachusetts. We have a fair amount of intellectual property around our microbial conversion protein technology. And we have developed an industry-leading functional single-cell protein that we are focused primarily and exclusively on aquaculture, solving some of the very most important big issues within the sector. I like to say more broadly and generally that there's a tremendous opportunity for biotechnology within aquaculture in an environment where the ability to make, produce, read, copy, synthesize DNA is moving at a rate something like 5 times faster than Moore's law. We have a tremendous opportunity here, but also just in time. When you have something like several hundred species in aquaculture, very new industry, only maybe 50 years in development. Compare that to something far more established like cattle, which has had millennia to develop with only one species of focus. You really need this fast, dynamic, powerful opportunity that biotechnology offers. Then on top of that, throw the opportunity to convert multiple feedstocks into these types of proteins. We really focus on liquid feedstocks as an opportunity, as a relatively easier and lower cost of conversion technology. So ultimately, these types of technologies, I would classify them more as a smart protein as compared to perhaps terrestrial proteins, because they are really tailorable through our technology, being able to deploy some really important functional molecules and enhance and grow in time to develop even further. So NipBio's first product to market is within the land-based systems. Feed is a critical component here. Water quality is absolutely paramount. And so to have a feed that works within these systems is a real critical issue. And we've developed some unique applications here that we'll be launching this year, in fact. So stay tuned and look forward to hearing more about that in the coming months. And RAS in general is a very exciting sector. Hardly a day goes by where you don't see a headline in the newspaper. Here's a selected sample from the 4 weeks mostly of February, where you see these events fall into several categories, including expansion and growth. There's a lot of good investment going into the sector, including this past section here alone, about $800 million. And you're really seeing a wide range of players, including existing net pen farmers starting to move into this sector as well. And then, of course, a lot of this is being driven by technologists in general. So exciting sector, and NipBio's commercially ready technology is going to address some of these biggest problems. We've scaled up commercially to date. We have over 40 trials under our belt. And we are the first of our kind to get a FDA approval for both finfish and crustaceans. Behind the RAS product, we have applications in juvenile feeds. Here's a study on the left, just recently completed for Asian sea bass, also known as barramundi, where we see all of our inclusion rates of KBM or NipBio meal had statistically improved performance compared to that of the control. Also, here on the right is a study where more nitrogen being converted into animal biomass and less into fecal pollution. Both of these lead to better, more robust adults, starting with improved juveniles. Also really important, we've seen time and time again, improved survival. Here's a study from Southeast Asia where early mortality syndrome, a bacterial-derived disease, which is really potent, can drive off 80% of your crop in a matter of days. We took some of our diets and we saw greater than a 2-fold resistance compared to the control diet. So there are immunonutritional benefits to our protein beyond just an amazing amino acid profile. But I'd like to take a moment with the last minute here that I have to bring your attention to some pre-competitive issues that all of my fellow panelists today are going to have to wrestle with. For us to really get into the market and bring our solutions in a big way to solve these very big problems. We're all working with different organisms that inherently has a very fragmented and dividing type of effect. Things that we need to understand better is, as NipBio, for example, we're going to be a 10% inclusion rate. How do we interact with other ingredients? And that's exactly what the Feed Innovation Network should help us with. In terms of regulatory, here in the US, one falls into 2 major categories, essentially permitting for offshore, which a lot of stakeholders have come together and formed the so-called Aqua Act. But we have a long distance to go with novel ingredients. And some of my fellow panelists have attempted in the past to put together a more unified voice. We still have work to do there because there is definitely going to be some barriers to adoption if we don't solve these very real issues. So appreciate your time. Look forward to your questions in the question and answer session. And thanks very much.
Thank you, Larry. Appreciate that. Good to see you again. So our next speaker is Mike Chichi, who's the Global Pathfinder for the Scholar Company. Mike has a diverse background in agriculture, working in everything from agronomics to food science and technology to dairy markets. He's been with Scholar Company since 2000, where he uses his expertise to expand the company's market share of functional proteins, fats, and oils into global animal feed markets. Scholar Company is currently launching a new alternative protein product with production scheduled to begin this summer and 3,600 metric tons of product expected in the first year. Let's hear from Mike about Scholar's work in the aquafeed space. Thank you, Mike.
Good morning, good afternoon, and good evening all. I am Mike Cece from the Schooler Company, and I'd like to thank you for the opportunity in allowing me to introduce to you Schooler and our novel protein ingredient, barley protein concentrate. Schooler is a 129-year-old, privately held, family-owned company based in Omaha, Nebraska. We have over 1,000 employees globally, located throughout North America and Asia. We consider ourselves to be solution providing experts of supply chain management and feed ingredient sourcing. We have many facility and storage capabilities and production facilities, which those production facilities are mainly located in North America. Schooler has about $4.5 billion US in turnover, and we are typically listed plus or minus 100th place on Forbes list of large privately owned companies. Here's our global network. As you can tell, many facilities in North America and in Asia, the Myanmar facility, along with trading and customer service offices in Indonesia and Shanghai, China. These are the markets that we serve, but again, we are solution providers for ingredient and feed manufacturers worldwide. Thank you very much. That concludes the introduction to Schooler, and now I'd like to introduce to you the barley protein concentrate as a substitute complementary protein to fish meal. The barley protein concentrate plant will be located in Jerome, Idaho, and is currently under construction. Will be commissioned in August of 2021. Why Idaho? Because Idaho is the largest producing barley state in the U.S. that will provide us with consistent supply and quality. The barley we source is non-GMO, and in that mountain region of Canada and U.S., annually is 13 million metric tons of barley harvested. And also the barley we are sourcing and originating is feed barley and will not compete with human consumption barley such as malting barley. The barley protein concentrate process is that we take the whole kernel and dehull it, removing the fiber and anti-nutritional factors. It is then ground and mixed with water. Towards the end of that mixing, an amylase enzyme is added to fractionate and separate the starch from the protein. The starch is then converted into C6-glucose, which we consider to be barley syrup, similar to a feed molasses. And that barley syrup is separated via centrifuge. And the protein continues on and is dried using a low temperature rotodisc steam dryer, similar to those that you see in fish meal plants, which many of you may know as a rotodisc dryer. The barley, after dried, is then packaged. The entire barley kernel is used. Nothing goes to waste. The fiber in the barley syrup, along with the screenings, goes to the local livestock feed, and the protein is utilized in aquaculture diets as a substitute or partial replacement of fish meal, the same that happens in pet food. In aquaculture. So how effective is it in aquaculture? So the barley protein concentrate, on the left side, you can see the typical analysis based on dry matter basis and as fed. The protein will be a minimum of 60%, but as you can see, we have fat, moisture, ash, phosphorus, and crude fiber listed. On the bottom left, you'll see the typical amino acid, a nice, nutrient-dense, well-rounded amino acid profile. The physical characteristics are that the color will be a brown color, and the granulation will be more of a crumble. The odor will be the aroma of a cooked cereal, and packaging will be in super sacks and bulk, and the minimum storage life will be 12 months. Now in the bottom right, we'll see a chart based on that will show you about phosphorus digestibility or bioavailability. And the barley protein concentrate is compared against whole grain barley and wheat along with menhaden fish meal. And the barley protein concentrate is showing a 22% higher availability than the Menhaden fish meal, which is a nice benefit in the RAS systems, the raceway systems, and the pond systems, any enclosed water system, which will be a benefit to aiding in water quality improvement. On digestibility, a trial was done at the Bozeman Research Center to measure the apparent digestibility coefficient, and the barley protein concentrate was compared to 3 very well-known and popularly used vegetable protein concentrates: corn gluten meal, soy protein concentrate, and soybean meal. And it was tested on 2 different salmonids, Arctic char and Atlantic salmon. And as you can tell, the performance was very good and very competitive with those 3 very well-known and used protein concentrates. Here's a palatability trial done with Marabini that'll show a decreasing level of fish meal and barley protein concentrate, replacing the difference up to 30%, and you will see that soybean meal has increased a couple percent. But this whole study, this whole trial shows that there really was no palatability issues. In fact, the feed conversion seems to have improved. So we've had some really good results on the coho salmon. Another commercial trial was done in the U.S. with Clear Springs Foods, now known as Riverence, on freshwater trout. Now this was a 2-year study tested on 375,000 trout, freshwater trout, from fry to harvest at a 30% inclusion, but also on the same farms you had alternating raceways where the control diet was used, and that control diet had included a high protein, highly digestible, nutrient dense fish meal. And that harvest, all the fish were then marketed to restaurants throughout the U.S., which were customer restaurants of Clear Springs. Now, the report that we received back from Clear Springs shows that there was no growth difference, no weight difference, and no conversion efficiency difference to the control diet. But another important feature is that the flesh color or the flavor of the fish meat did not change at all. Very nice results, and we're very proud of that. So that ends the introduction to Schooler and also our barley protein concentrate. Thank you very much, and I will be available for the Q&A session.
All right, thank you very much, Mike. We appreciate that. Our next speaker is David Say. David is the CEO of NOVONUTRIENTS. David launched the Aquafeeds Fund in 2004 that includes a 5-company seed stage portfolio focused on aquaculture nutrition, including Open Blue and Ovasi and Fortuna. The companies he has been involved with have raised more than $250 million. In 2017, David became CEO of NOVONUTRIENTS, which transforms CO2 emissions into food and feed protein ingredients. Just last month, NOVONUTRIENTS received regulatory approval in Japan for use in all animal nutrition, including Aquafeeds. NOVONUTRIENTS has also fulfilled most of the F3 Feed Innovation Network's ingredient evaluation workflow tests. And now we will hand it to David to tell us more about NOVONUTRIENTS. David, go ahead.
Hello, I'm David Say, and I'm the CEO of NOVONUTRIENTS. We make food and feed from CO2. So there are 2 global problems that NOVONUTRIENTS is involved in solving. The first, which would be familiar for this webinar, is the matter that there's really too little protein relative to growing global demand. And then the other is that we're putting way too much carbon into the atmosphere and creating climate change. Our inputs are really at the core of what differentiates our technology from other single-cell proteins. We primarily rely on carbon dioxide from industrial processes. We pair that with hydrogen, our energy source, So these are both gases which we can use in their least expensive and most plentiful forms, which are when they're not pure gas streams, but rather when they're mixed with other gases, which is what tends to happen when you're actually out there making stuff in the rest of the economy. And so along with a few other things, that becomes the aqueous media for our fermentation, which is based on bacteria, elements of our technology that are in the hardware, in the physical systems, and others that are in the biology. And the result is transforming the circular economy inputs into protein products, things that can go into formulated foods and feeds. So here in yellow, we show where carbon dioxide is primary, in blue, where hydrogen is primary, and in green, where you find both in meaningful amounts. And so these are the sectors of particular interest to co-locate our facilities. We have long thought that aquaculture is the first and best market. It offers moderate and strong margins. The value chain is really ready and needs these types of novel alternative proteins, as is demonstrated by the existence of this webinar. And the market size is solid. This is the area where we've also made the most regulatory advances, having recently received approval for animal nutrition use, including aquaculture in Japan. We call our product a super prime protein. It has all the amino acids. It's a complete protein. It's 95% digestible, highly palatable, 73% crude protein level on a dry basis, and also contains some rare and valuable additional components, hemes, vitamin B complex, carotenoids, naturally occurring here. These are first feeding screenings with U.S. Fish and Wildlife Service, where we showed comparable growth and survival in rainbow trout to the control diets, which had gold standard and high levels of fish meal. The other product family that we will be producing are our what we call our novoceuticals, which are really food and feed additives. So whereas the bulk proteins, while high performance, are natural and done without any genetic modification, when you want to produce specific biochemicals in significant amounts, as we show here for canthaxanthin and more generally for a category called carotenoids, then you use genetically modified workhorse to do that production. In our case, that means we can use the same feedstocks and the same fermenters that we do for our protein, but just a different downstream processing. So looking at the macro trends, there are three that we think are particularly favorable for us that are underway and will continue to accelerate. The two that may be new for this audience are the decreasing cost of hydrogen. And this is true whether we're talking about green hydrogen, gray, blue, the kind of byproduct hydrogens that we're looking at, or for the net cost of carbon capture, which is to say both the cost of capturing the carbon itself, as well as any government interventions that may affect that from a market perspective. Our pilot systems, as we scale up, will look something like this. This is an early rendering here on the right of a 1,000-liter system that would produce more than 1 ton of bulk protein a month. Subsequently, new designs have physically compressed this, so it's just more clear how it can be containerized and easily transportable. Our model is to arrange finance for licensees who actually would operate the manufacturing plants, the arrangements with the CO2 emitters who might themselves be providing additional finance, and then also help put in place the contracts, the long-term purchase agreements with the end users, the folks who are actually formulating aquafeed, feed, or food products. At the moment, we have our first technology licensee that spans the first 3 segments of this chain, from project finance through CO2 emission to holding a license. And that's a quicklime company in Japan that has contracted to work with us through pilot, demo, and commercial phase. We also have support from Chevron through its Catalyst Grant program as another major emitter. And on the customer side, public collaboration with Skretting, which is explicitly an evaluation for potential offtake agreement. As we move through these pilots, we start to see commercialization for our high-value products, initially in Japan, then moving on to a chain of larger projects in the Gulf Coast where there should be particularly good economics, and then more and more larger and larger projects on a global basis after that. Among other things, this allows us to get to a very significant level of carbon dioxide upcycling. By 2030, we should be doing between 4 and 5 megatons a year based on some relatively conservative growth curve. Hopefully we can do better than that. You'll achieve that sooner and grow even faster afterward. So that's how we intend to mitigate the fact that there's too much carbon and solve, or be part of the matter of solving the challenge of too little protein. Thank you very much.
Thank you, David. Appreciate that. Lots of good information there. Okay, next up we have Alan LeBlanc. Alan is the Vice President for Callista's Aquaculture Business Unit. Alan joined Callista in 2013 and is responsible for global market and sales strategy. He previously worked in venture capital at Ballast Point Ventures and investment banking at Raymond James. Callista is a biotech company whose aim is to make more from less by fermenting natural gas to create new food products. FeedKind is Callista's new non-GMO protein that has been commercially validated through trials in aquaculture, agriculture, and pet food. Callista currently operates a pilot plant in Teesside, England, where they produce about 50 tons per year. They're in the process of building a 20,000 ton capacity plant in Chongqing, China, and it's expected to go into operation next year. So let's hear the latest news from Alan. Alan, good to see you. Go ahead.
Hello, and thank you for having me. Thank you, Barbara and Kevin, for having both myself and Callista and FeedKind on. By way of introduction, my name is Alan LeBlanc. I work at Callista. I lead up our aquaculture group. We've been growing rapidly lately. We've got a technical director in China now, a technical director in Thailand, and hopefully we'll be adding some additional team members around the world. So today I'm going to do a quick introduction to FeedKind and the company. So we're a single cell protein company, like I think everyone else is here. But the thing that sets Callista apart and really differentiates FeedKind is obviously the product, but how it's made. So we're using a gas fermentation. We've got a microbe, a Methylococcus. It's present in soils and swamps all over the world. We're using its natural form, putting it in a fermenter, feeding it natural gas, methane, oxygen, and keeping it in a happy environment and harvesting that biomass as the product. And so the thing that sets that fermentation process apart, and gas fermentation generally, is unlike a sugar where you drop it into the water and it dissolves very easily for the microbes to get it, the gas tends to separate. And so we have a proprietary fermenter, which has a lot of engineering in it around mixers and gas injection to really help those microbes get to the gas and continue to reproduce and multiply. So we have a pretty simple process. Post-fermentation, it's centrifugation, dewatering, spray drying, and we have a powder or pellet that we then sell. So where we are as a company is kind of an exciting point in that we're at the commercial scale up. So we have a plant in the UK that's a pilot plant, produces around 50 tons per year, has a capacity of 50 tons per year. And we're building a plant in China with Adissio that will produce 20,000 tons per year in about 18 months, so Q3 of next year. It's under construction as we speak, and the engineer's rendering is over here on the side of the screen. But on that, I mean, we've partnered with Adissio. So Adissio, if you don't know them, they're a major methionine producer based in France, and they're owned by Blue Star ChemChina in China. And so we've formed a joint venture with them to really commercialize FeedKind, both manufacturing and sales throughout Asia, with an initial start in China. I mean, it is the world's largest aquaculture market, and it's the place where we think we can make the biggest difference, both impact on the industry as well as obviously growth and sales for our company. So when it comes to sustainability, single cell protein, but especially gas-based proteins like feed kind, where they don't use agricultural inputs, really stand out. So on the left, you have some greenhouse gas profiles of feed kind and different protein choices. But in general, within the ingredient space, feed kind's kind of like right in the middle of the barrel for greenhouse gas emissions. So depending on where your soy is from or what fishery you're in, it really, is right in there with fish meal and soy protein concentrate from a CO2 emissions point of view. But where it's completely different is in freshwater use, land use, and really eutrophication. So because we're in a fermenter, all of the phosphates that are going in there, all of the ammonia, the nitrogen nutrients are getting consumed. Whereas in a field, you have significant runoff, as we all know. And similarly, we're in a plant that's maybe 10 hectares and producing up to 100,000 tons of feed kind. And you compare that to what you need for soybeans, it's 2,500 square kilometers, which is an area the size of Chicago. And as the company grows and as our production grows, I think we're really going to start to make a major difference in land use needs. And it's something that I think the industry is starting to talk about. There have been some great articles by GAA around soy protein and how it's been a great growth enabler for the industry so far. But what happens next? Can it really continue to grow if there isn't that land available for growth? So what's the industry looking for in a single cell protein? Of these, I think the one that I think is most important and really sets feed kind apart is high density. So feed kind is 71% crude protein. It's 60% amino acids. It's got all the essential amino acids. And so that, to me, is what enables the feed company to use it easily. The ability to fit it into a formulation that might have high nutrient requirements or very little room, in the sense that they don't have a lot of space for fiber or ash where it might get in the way of producing an effective or quality pellet. And that's really where feed kind sets apart and can really work in lots of different applications and can help make feed formulators and feed manufacturers' lives a lot easier. And the second one I'll focus on is really the scalability. I mean, we're doing that now. Our 20,000 ton per year plant in China will be the largest single cell protein production facility in the world, and it'll also be significantly larger than the insect manufacturers that are producing today, who tend to be on a protein basis, in the 3,000 to 5,000 tons per year range. So we're very excited about that scale and kind of bringing that scale to the aquaculture feed industry, which really needs it and requires it before they adopt new ingredients. So Barbara and Kevin asked me to show some data, and I did want to do that. I didn't want to overwhelm it because I know we don't have a lot of time. So I focused on one that I think is relevant to this discussion and also relatively recent, in that we just published this the end of last year in aquaculture. But it's work we did with Kindai University in Japan, focused on hamachi or seriola or Japanese yellowtail. And for us, it was valuable and exciting because it's a fish and a species and an application that has really been resistant to the replacement of marine protein with vegetable proteins. The commercial feeds today still are 35 to 55% fish meal. There's very strong enteritis effects from high vegetable protein content. And there's also a challenge around palatability, where the fish will, if it doesn't taste, feel, look right, the fish will not eat it readily. And so we did 2 trials with them, but the formulations that I'm showing here to the right are the second one, which was about optimizing the feed kind inclusion level for the yellowtail. And what we found was really the 17 to 20% of total feed gave us great feed efficiency, great growth, and the same animal health as we saw on a marine-based diet. And for us, being building a plant in Asia, in China, and focusing on a lot of these carnivorous species that have less well-defined nutritional requirements, this was a great template for us and something that we're excited about and excited to see published. If you're interested in the paper or more details on the results, I would be happy to share this with you. And the last thing I'll focus on that really, I think, differentiates feed kind from single-cell proteins generally is the ability, the traceable nature of it. So we can really track the product from when it's made to in a feed to in a fish. And as companies and supply chains become more integrated and more focused on provenance and sustainability, I think this is really enabling for a lot of brand owners and retailers to talk about a seafood product and have the traceability features built into the product itself and not on a label or a QR code. So with that, thank you for my time and look forward to answering questions later. Thanks. Bye.
Thank you, Alan. That was great. We appreciate it. And we'll look forward to you in the question and answer here momentarily. Next up, we've got Mark Rotman. Mark is the CEO of iCell Sustainable Nutrition. Mark's a seasoned executive leader and entrepreneur with over 30 years of global management experience. He currently serves as CEO of iCell and was previously an executive at GE Water. iCell utilizes a patented process to clean effluent water from the food industry and feed the leftover organics to microbial biomass, resulting in clean water and single-cell proteins that are suitable for the feed industry. iCell currently produces thousands of tons of single-cell protein products and commercializes them in the China feed market. We're excited to hear more about this process from Mark. So Mark, please go ahead.
iCell Sustainable Nutrition is a Hong Kong-based water and protein technology company which currently operates 3 facilities producing single-cell proteins in Asia. Initial production began in 2014 with its first site adjacent to the Anheuser-Busch facility in Qiantai, China, and has since expanded to treat food water effluent from sugar and seafood processing sites. The iCell business is commercially proven and has produced and sold tens of thousands of tons of single-cell protein-based products since inception. iCell has set an aggressive expansion plan around the world and has been selected by Quality Salmon of Sotenäs, Sweden, one of the largest salmon aquaculture projects in the world, to be the water and nutrient recycle provider for the site. Additionally, iCell has partnered with Paradis Capital and multiple aquaculture companies in North America for integrated land-based RAS projects. iCell, the future of sustainable protein production, is already here today.
iCell Sustainable Nutrition, the solution for water and nutrient recycling in the food, beverage, and aquaculture industries. Water is a large processing component in the food industry. And that water often picks up the bits and pieces of the food product it has been washing. When this happens, it can be very expensive to clean this water before discharging back to the environment. Land-based recirculating aquaculture systems, LB-RAS, are the future of sustainable seafood production. And yet one of the largest issues they face is dealing with all the water that is needed, both from a production standpoint and a regulatory discharge standpoint. Water that must be kept clean for the fish and shrimp to grow, and water that must be cleaned before sending it back to the environment. And that is a costly problem. iCell brings a unique and patented solution to LB-RAS. The ability to use existing water from food processing industries as both the recycled water source and an environment to grow a natural protein ingredient. How? First, the water is cleaned by iCell through a fermentation process whereby iCell's microbial proteins eat the organic matter in the water. When this happens, the microbes are cleaning the water of the organic material. This source water can actually be from most any type of food processing industry: seafood, poultry, pork, food or beverage. After this microbial biomass is grown, it is then harvested, dewatered, and dried into a single-cell protein, SCP, ingredient. This SCP is then used as an ingredient in the feed formulations of shrimp, salmon, trout, and other aquaculture species. It can also be used for high-growth land animal species such as poultry, swine, and beef. The iCell solution is not only sustainable, it is carbon negative. How? Without the iCell process to convert the nutrients in the food processing water into a feed ingredient, those same carbon-based nutrients end up decomposing and converting it to methane, a greenhouse gas which is 25 times more powerful than CO2. Protein usually requires a lot of land and water. An iCell Aqua site can produce 500 metric tons of protein on 1 acre of land versus the 350 acres needed for the same amount of soybeans. And it can produce this protein with less than half the energy required for other protein ingredients. Every 1 acre iCell Aqua site is like removing the carbon from 10,000 cars from the road. iCell brings together 2 water processes which are normally considered separate: seafood farming and seafood production. iCell integrates them such that the result is the conversion of what would have been waste organic matter into a valuable protein feed ingredient, and at the same time creates a recycled water stream. iCell Sustainable Nutrition, cleaning water and creating single-cell protein, SCP, feed ingredients.
Scaling is really the next step for the iCell business, and producing SCPs can be capital intensive. Money for scaling and sites must come together at the same time. From a funding standpoint, we see the market is flush with cash, but it's looking for big ticket investments in the tens of millions and not just 5 million here and there. So we've taken a new approach by integrating iCell with aquaculture. We create a business model with more synergy and larger investment sizes in the $30 to $50 million per site range. And really this leans towards iCell's strength, because in this synergy of solving food wastewater issues for a food company, producing SCPs so that we create a valuable product, and then using that newly cleaned water for the SCP and for the aquaculture, we create a cycle that's just wonderful for the environment and for the business model. In 3 years, we see ourselves with $100 million of deployed capital and systems, many SCP sites operating in China with tens of thousands of tons per year of SCP production, and then in North America, Europe, having 3 or 4 of these highly integrated aquaculture sites where we clean water, make SCPs for thousands of tons of aquaculture per year.
携带经过几年的发展，目前有多个稳定运行的规模化工厂，如我们经与百威英博、星光糖业、正大等客户实现长期战略合作，现每年有数千吨的单细胞蛋白饲料销售给我们的客户。携带不同于传统的饲料生产工厂，我们的原料在别人看来是废弃的、没有价值的，生产出可媲美鱼粉、是具有成本优势的生物蛋白，也是可持续的新型蛋白源。携带有成熟的运营和研发团队，已对全球100多个规模化食品工厂客户完成了技术评估，都可实现有机废水资源化生产单细胞蛋白。利用我们独有的 M42模块化生产系统，未来可实现项目的快速复制，并且大幅降低投资和运营成本。单个项目的 M42系统投资金约100万美元，3年后将有30个以上的项目实现运作，生产出数万吨的单细胞蛋白产品。
SCP 的生产过程不与人争地，不与人争粮。与动物和植物性蛋白相比 ，SCP 适口性好，富含蛋白和细胞活性成分，同时不具生物安全隐患和抗营养因子。目前，协赛已成功生产并销售几千吨 SCP 进入动物饲料和养殖领域，包括畜禽、水产、反刍和毛皮动物等。随着协赛技术的进一步规模商业化 ，SCP 的生产成本将大大降低。同时，协赛也正围绕 SCP 做一系列的纵向开发和横向衍生产品的研发。SCP 是一种安全、优质、环保、经济的蛋白资源。SCP 将成为解决人类蛋白需求、续命增长的有效途径之一。
Thank you, Mark. We appreciate that. And next up, we've got Paul DePoe, who's the Business Development Director for Feed for Unibio. Paul has worked in sorry. Paul has worked in ingredients and feed sector for the last 35 years and has a background as a bioengineer. He has been with Unibio since 2019, where he's responsible for the business development and sales of the product Uniprotein for animal nutrition. Unibio has microbial fermentation to convert any natural gas or methane into high quality somebody Unibio has microbial fermentation to convert any natural gas or methane into high quality protein for fish and animal feed. Unibio's Uniprotein product has been approved in the EU for animal and fish feed, and is certified organic by organic farmers and growers in the UK. Now we can hand it over to Paul to hear more about Uniprotein.
Hi everyone, my name is Paul de Pauw and I'm responsible at Unibio for the business development of Uniprotein in the animal nutrition sector. Uniprotein is a protein and fat-rich biomass, 70 and 10% concentration respectively, produced through fermentation from methane. The concept saw light in Norway in the 80s and has been turned into a cost-efficient industrial process by Unibio through the development of the Europe fermenter. A lot of natural gas is needlessly being wasted as a byproduct from drilling activities all over the world. The Unibio fermentation process will allow this methane to be converted into a nutritious biomass instead of burning it into CO2. This, together with economic water and land usage and no discharge of nitrates into the environment, provide Uniprotein with an excellent sustainability score, which is monitored ongoingly through life cycle assessment. Furthermore, this process lends itself to be integrated into a circular concept in which biogas produced from agricultural and other waste is feeding a fermenter that in turn produces a biomass for animal or human consumption. A unique combination of features renders Uniprotein into a versatile raw material for usage in a broad range of feed formulas. Firstly, the high protein density and a rich essential amino acid profile make it an ideal ingredient in aquafeeds for salmonids, shrimp and carnivorous fish culture, creating space in the formula to include other less concentrated raw materials. In addition, the fat is bound to the protein, which benefits the processing of high fat containing feeds. Several studies done in juvenile rainbow trout, salmon, sea bass and tilapia have shown that Uniprotein can replace fish meal without loss of performance. In tilapia, for instance, 100% of fish meal, which was 25% of the protein, was replaced. And in a 1-year salmon trial, up to 19% Uniprotein was fed, again replacing fish meal. We measured the in vitro true protein digestibility to be at 85%, and in a recent digestibility trial done in Chile in salmon, Uniprotein was found to have no significant difference with fish meal. Secondly, Uniprotein is a very clean protein fat source. No antibiotics, no fertilizers, no heavy metals, no mycotoxins, no pesticide residues enter the process. The absence of such contaminants is of particular importance in feeds for sensitive animals or life stages, like starter feeds for piglets or calves, or in pet food. A 1-year study was completed recently in Denmark, in which more than 3,000 piglets were given a feed with up to 10% Uniprotein inclusion, replacing fish meal and potato protein with an equally positive performance as the reference. Finally, Uniprotein is non-GM and also a certified organic, which gives access to a new large volume raw material to producers of organic feeds who are facing tightening regulations in the EU, excluding conventional ingredients. The organic sector is a strongly growing one, and we intend to take a major place in the supply to that segment. So, what stage of development are we in? Well, I'm happy to announce that the first industrial ULUP fermenter has recently started up in Russia, near St. Petersburg. The initial capacity will be around 6,000 tons. It's a modular concept, and additional ULUP modules will be added in the coming years. So if any of you listeners are looking for product, don't hesitate to contact us. For EU-based companies, I add that Uniprotein is a registered feed material in the EU, and that the GMP certification of the Russian plant is expected by September. The obtention of GRAS status in the US is in progress. The Russian project is the first in a row. Other licensing and co-investment projects are in various stages of development, and in 3 years, we expect to have several more production plants up and running across the globe, producing more than 100,000 tons of Uniprotein per annum. The development of the technology is an ongoing process, of course, and we continue to improve that in our R&D facilities in Kalundborg, Denmark, where we have a demo-scale ULUP fermenter in place. We're also continuing our process and product R&D to broaden our portfolio with products that are more suited for specific animal species and the range of derivatives for human consumption. The first one in the pipeline will be a product with reduced nucleic acid content, which will greatly increase the access to the pet food market. Unibio is looking with great expectations to the future. I thank you for listening.
Thank you. Thank you, Paul. We appreciate that. Okay, next up, we've got Dr. Peter Williams, and Peter is a senior nutritionist for Green Plants and Fluid Kip Technologies. Peter has a background in animal nutrition in both academia and commerce. He's currently the CEO of Ag Bio Limited, which is an animal nutrition consultancy focusing on alternative proteins for food and feed. He's also the senior nutritionist for Green Plains, is a leading biorefinery company and innovative producer of ultra-high protein and novel ingredients for animal and aquaculture diets. Green Plains currently produces over 500,000 metric tons of their corn protein concentrate product each year, with plans to increase to 1 million tons per year by the end of 2023. So Peter, please, can you join us?
Hello, my name is Pete Williams. I'm senior nutritionist with Green Plains. I've been a livestock nutritionist for the last 40 years, and for the last 10 years I've been focusing on alternative proteins. Today I'm going to tell you about a real success story, corn fermented protein. Now, corn fermented protein is produced by Green Plains. Green Plains is a leading agri-tech company. We're committed to transforming our platform to process annually renewable crops into more sustainable, high-value ingredients. Green Plains' vision is to take a traditional dry grind ethanol plant and turn it into the biorefinery of the future. We are focused on developing and utilizing the fermentation process to produce agricultural and biological products using renewable crops and turning them into sustainable, value-added products. Our extensive current asset base, exclusive partnerships with Osprey and BlackRock, and the recent acquisition of Fluid Kip Technologies create a very powerful ag-tech opportunity. The biorefinery opportunity currently encompasses the ultra-high protein, which is the topic of this presentation, but also renewable feedstocks for advanced biofuels in the form of corn oil, high purity specialty alcohols for use in cleaners, clean sugar and the production of glucose and dextrose, and finally, carbon capture and the sequestration of CO2 to aid in making the whole biorefinery process carbon neutral. Corn fermented protein is the subject of the talk today. Corn fermented protein, or CFP, is the term submitted to AFCO to describe this alternative, grain-based, high-concentration, functional protein. Currently, the product is produced from a corn ethanol fermentation, which produces a 52% protein content product. The process to produce CFP is a completely mechanical separation process. This process has been developed over the past 10 years by Fluid Quip Technologies. Fluid Quip Technologies have vast experience in the wet grind process. The process to produce CFP is a completely mechanical separation process using centrifugal forces and sequential washing to produce the product. So the product is pure corn protein plus a high proportion of the yeast generated during fermentation. There's no change in the water balance of the plant. There's no additional water required for the process. Since the product is already classified in the AFCO handbook under distillery products, there are no regulatory issues concerning corn fermented protein. The product is regulatory compliant in international markets. We're currently producing a 52% protein product from corn. The digestibility of the protein, measured in vivo in Atlantic salmon, is greater than 95%. We've measured ileal digestibility in poultry and it's slightly lower at 90%. Lysine availability is high. We use benign drying using the ring dryer, and using the carpenter lysine technique, our lysine availability is greater than 99%. Fat content is 4%, crude fiber 7%. The functional characteristics of this protein come from the 24% of the dry matter yeast, which is spent yeast from fermentation, which gives us the yeast beta-glucans and mannans, which are useful for immune stimulation. The product specification, based on a corn-based product, is 52% protein. With development work, we hope to raise this to greater than 60% crude protein. Ileal digestibility, measured in vivo in Atlantic salmon, is 95%. We've also measured ileal digestibility in poultry. 24% of the dry matter of the product is spent yeast from fermentation. This spent yeast provides yeast beta-glucans and mannans, which are implicated in immune stimulation. What are the target species for this product? Corn fermented protein is a benign, highly digestible, concentrated alternative protein. So there are lots of opportunities for the use of this product. We've replaced fishmeal and soya isolates in aquaculture. We've carried out over 30 trials in both academic and commercial facilities testing the product. We've done trials in carp, tilapia, trout, shrimp and Atlantic salmon. And without exception, based on commercial formulations, we've either achieved the performance of the commercial formulation or bettered that performance when we've used CFP in the diet formulation. We've replaced soya isolates in diet for turkey poults. We've replaced soybean meal in feed for broilers. We've replaced protein supplements for starters for pigs. And the protein has some particular characteristics that make it an ideal protected protein for dairy cows. There's a wide range of target species, but our current focus is using this protein as an alternative replacement protein in aquaculture. All this trials data is available for review if required. One of the unique characteristics of corn fermented protein is that it is already commercially available. Commercial production of corn fermented protein shows that it's a consistent product. It's produced in commercial volumes, with annually producing over 500,000 tons per annum from 6 plants in production, increasing to 1 million tons per annum by the end of 2023. With 6 plants in production, we have resilience in the supply chain. We have redundancy in the supply chain. The challenge to us now is to roll out the technology globally. Investigations are already in progress with European plants. If it was a European plant in production using non-GM grain, then this corn fermented protein product would be a non-GM alternative protein. Perhaps the most important unique characteristic of corn fermented protein is that it is currently commercially available. There's a tremendous future for corn fermented protein. The future for corn fermented protein is our partnership with Novozymes. With Novozymes, we're undertaking continuous product development. Our first target is to increase the protein content to greater than 60% using enzyme technology. Then we will turn our attention to improved characteristics through changing the composition of the yeast. When we can recover the yeast, we can get the additional benefits through the yeast components. There's an ongoing target of removing all anti-nutritional factors. And finally, direct CO2 sequestration from the plants producing CFP may allow us in the future to produce CFP in a manner that is totally carbon neutral. So corn fermented protein is an exceptional alternative protein for aquaculture. If you'd like more information, then please don't hesitate to get in touch with us. You'll find all the details, contact details on the slide here. Cheerio and goodbye.
Thank you very much, Peter. That was very informative, and I'm sure we're going to have several questions about that. What I'd like to do now is if all of the panelists could join us, and we just want to run through, have everybody briefly introduce themselves so that our folks can put names to faces there on the panel. So please, let's go ahead and, okay, we've got everybody up. Peter, why don't you go ahead since you were just up?
Well, good evening, good morning, everybody. It's been an early morning for me. It's been a fascinating introduction this morning to these new alternative proteins. It's a very exciting area to be working in, and we're all pushing along in the same direction. So we believe with using the infrastructure of the dry grind ethanol plant and now producing a designed product out of the ethanol plant rather than the co-products of the past, that we can really contribute to the new proteins of the future. So over to my co-patriots.
Thank you, Peter. Mike, you're next on my screen. Mike PC, please.
Yeah, thank you very much for joining the presentations and the webinar. The barley protein concentrate, I think, is a very nice alternative protein choice compared to a lot of other cereal grain type proteins. It's a low input requirements for low water use, low fertilizer, chemicals, pesticides, along with being non-GMO based. And I think some other benefits would be, compared to other type of vegetable protein concentrates, does not promote any deforestation because everything is produced on current production acres. So it's a great alternative to look at for fishmeal alternatives, fishmeal extenders. And again, if you have any questions, please get a hold of me. But thanks for joining.
Thank you, Mike. David, you're next on my screen, please. David Say.
David Say, CEO of NOVONUTRIENTS. We're really focused on scaling up to achieve superior performance at a lower cost than alternatives. And so that's really our focus.
Thanks, David. Good to see you again. Alan, you're next up, please.
Hi, everyone. Alan LeBlanc. Thanks for having me. I won't add to anything at this point.
Okay. Very brief. All right. Thanks. Paul DePoe, you're next, please.
Paul, I think you're on mute.
Yeah, Paul, you're on unmute, please.
Hi, everyone. Paul DePoe, Unibio, responsible for the sales and development of animal nutrition of Uniprotein. An exciting webinar. Like everyone, we are looking forward to the future, because single cell proteins is the future for animal nutrition, and I think we are going to play, or we hope to play, an important role in that. So I'm excited for it, and I'm we're interested to hear questions.
All right, thank you very much, Paul. Mark Rotman, please.
Hello, everyone. Mark Rotman here. I'm in the Philadelphia, Pennsylvania area, joining the call. Thanks for your time. Nice to meet you. I know Mike CC and David Say from the past, so good to see you guys. And I'm actually probably want to make a group comment that is, I think we're all in this together. A lot of people say, who's your competitor? And our competitor is not one another, because we all bring a certain solution, and really sustainability is our competitor, and finding ways to just simply do better. So good to be with you all, and hope to answer some questions.
Yeah, thank you, Mark. Good point. I think I saw lots of nodding from everybody, so I think everybody agrees with you. Larry Feinberg, please. Larry, come on.
Hi, everybody. Larry Feinberg, CEO and founder of NipBio. Some really nice comments there. I definitely echo. Good to see some friends again. It's been a while. It's been a tough year, so good to be reunited. And thank you, Kevin and Barb, for bringing us all together today. And glad to be part of this panel. Thank you.
All right, appreciated that, Larry. And let's just jump right into the questions. And so a couple people had this same one for David. Say, you gave a good description of CO2 and hydrogen, but a couple people said, where's his nitrogen coming from to make proteins? So I figured we better let you answer that one.
Yeah, so look, of course, we need all the elements that are generally required for life: nitrogen, phosphorus, potassium, sulfur. But the idea is to use non-nutritional sources, to use inorganics. And specifically for nitrogen, our bacteria can use ammonia, nitrates, nitrites, and a variety of other nitrogen compounds. But ammonia is the default because of the availability. In certain locations, there may be better feedstocks, or at least a better value in feedstock.
Okay, good. Thank you, David. That helps. So we've got one for Larry. And for Callista, a couple of people had similar question. Your trial still had a pretty high % fish meal that you presented. And what additional trial, or do you have additional trials planned that are going to further replace the fish meal? Did I say Larry?
Yeah,
or Alan?
yeah,
okay.
That's right. I heard, yeah, I saw that question. I think it's a good one. You're right. So the trial we did is we replaced up to actually 75% of the fish meal in the first trial. And what we saw is that at some point you start to get a significant reduction in feed intake. And I don't think that's a feed kind specific attribute. I really think it's a lack of marine protein is what we're seeing. And that's something that we've experienced with varying species, but obviously the more carnivorous tropical marine species are the ones that kind of show it the most strongly. So yes, we're continuing to work with both palatants and formulators to try and increase that level. But I think, from our perspective, we don't have to get 100% of fish meal out of every formulation to build this industry and to help feedkind get adopted in the industry. I think it's really about taking out that incremental 5 to 10%, which traditional plant proteins haven't been able to do, and then growing from there.
Okay, and we've got another question here. Somebody asked about your Memphis operation that started. It wasn't mentioned, so
yeah. So that's a Cargill facility. We've licensed that production facility to them. So happy to get you in touch with Cargill to follow up with them.
Okay, good deal. Thanks. Okay, and so next one for Mark with iCell. Again, a couple related questions about regulations that prohibit salmon trimmings or other fish processing to be turned back into same species feed. So we're just curious about what are the regulations when you use your product, which isn't exactly trimmings, but still coming from similar species. So how does that get handled, Mark?
Yeah, so that question comes up a lot. And if you think about it, we're not feeding a salmon back to a salmon. We fed the salmon particulate in the water to a bacteria and harvest a bacteria. So a tomato is not the fertilizer, a salmon is not the sardine that it ate, and our bacteria is no longer a fish. It's a new organism. So that's really not an issue in terms of in terms of feeding back to the same species.
Okay. Great. That's what I think we're looking to hear. Okay, and the next question for Peter. Your product is currently being produced at a considerable scale. What advice do you have for other ingredient companies still working to achieve commercial scale production?
We've heard already this morning, scale, consistency, and resilience in the supply chain is one of the key attributes of any new alternative product. I've worked in the feed industry for many years, and when I talk to my compatriots and I asked them, how much do we need to produce to get bin space in your mill? And they turn to me and say, well, if you can give me 100,000 tons per annum and you can give me resilience in that supply chain, I'll give you bin space. So all these technologies, one of the biggest challenges that we all face is getting that volume that is required to be commercially viable. And I mean, we need the regulatory compliance, but even if you have the regulatory compliance and you can't find the volumes and scales that the industry needs, you don't get into the feed mill. So it is, to me, right at the top of my list as to what we can do. And we'll be 500,000 tons at the end of this year. We hope to roll out the technology across the dry grind ethanol plants globally. And that gives us up to the target that we're looking for at 100 million tons per annum. And then we become a truly commercial alternative protein. So volume is key.
Okay, thank you. Mike, for Schooler, we had a question here about positioning the barley protein, both in terms of nutritional, commercial value. Where do you plan to position this? Is it more for replacing soy protein or fish meal, something else?
Oh yeah, thank you for the question. I think with the barley protein concentrate, the product is an alternative protein to be used, along with a lot of the other proteins that are being discussed on this webinar. Every farm, every management system is different, and every environment is different. So I think it's a choice in the sense of finding which one works best for your system. It's a great choice, no doubt. We've run quite a few different feeding trials, some commercial, some just R&D, small scale, and it performs very well. And I think also with any concerns about sustainability, environmental stewardship, those type of things, the barley protein concentrate falls in very highly if there are those concerns within your production system or within your company. So I think answering the question, I think it's as we know, and through trials and testing different ingredients, you got to find out what works best for your system.
Okay, thank you. So Larry, another question for you. What are the regulatory challenges that fermented or single cell proteins are facing these days?
Yeah, it's a really important question, and it was encouraging to hear that several of the panelists have gone through various different jurisdiction approvals, but that's also part of the issue. one approval in Europe, not necessarily approval in the US, not necessarily the same as Japan, for example, right? And so in an ideal world, there would be some reciprocity with these different dossiers that are submitted that take many years in some cases to put together, formulate, and then go through the process. So it's a very clunky, very inefficient process that we have today, and it's, as a category, we're very disadvantaged compared to, for example, insects, where they're really all working on black soldier fly. Where we all have our favorite bug in this group here, and so it's about finding the commonalities and being able to sort of speed up the process, because frankly, and probably in all of our cases, we got through an approval on a product that was developed maybe 5, 6, 7 years ago in some cases, and so with all the promise of biotechnology and how fast and the ability to accelerate. But to have something that takes multiple years on the tailpipe to get approved, there's just a real disconnect between the potential of the technology and the practical adoption. So it's a hard one to solve. I know that many of my colleagues and I have been working on it in some degree for a while now, and we need to continue to work on that for sure.
Okay, thank you, Larry. This question, it was directed to David Say, but I think I'm going to ask a couple folks, because I think several of you have got similar products. So this is from somebody who's studying sustainability, and was asking if David, if you could share your circular feed model as that applies to circular economy. And of interest to people like the MacArthur Foundation and others. So could you just tell us a little bit about how you look at this as a circular feed model? And I'll ask somebody else after.
Yeah, I mean, I'll take some liberties with interpreting the question, because I'm not entirely sure what they mean. But, as far as how this fits into a circular economy, we're really talking about what are other essential industrial or energy processes that put out waste or byproduct streams, and then how can we use them? And so we need about 5 tons of CO2 and 1 ton of hydrogen for every 2.3 tons of protein-rich biomass, essentially product that we're producing. And so for us, if I can extend a little bit and talk about sustainability, the carbon footprint of our process really depends on the carbon footprint of the hydrogen. And so we'll really benefit from ongoing improvements in the global hydrogen infrastructure, particularly green hydrogen that can be generated through renewable power. But for today, we're interested in the lowest cost hydrogen that we can find, because our primary objective is to become commercial, and then achieve greater and greater sustainability over time as the underlying technologies improve outside of our company.
Okay, thank you. Paul, how would this apply circular economy to your operations? Oh, we need you on unmute, Paul, sorry.
Yeah, well, it depends, of course, on the source of natural gas or biogas. It makes a big difference. But the interesting thing of the process is that it can be linked up with a biogas installation. So you can have a situation where where you have a sufficient high concentration of agricultural activity, that you have a biogas unit converting all the manure and the waste, agricultural waste, into biogas, which is then fed into a U-loop fermenter and turned into an animal feed ingredient or human food ingredient. That's the ideal. And your question might be, okay, what do you do with the ammonia that you need into the system? Of course, you can use the biogas as an energy source to produce your ammonia and your oxygen on site, and so then you can turn it into a very, very low or a system which has a very, very high sustainability score. So it depends, but you can go you can be extremely efficient if you work on biogas. Ultimately.
Okay. Thank you, Paul. Appreciate that. I've got a question here that's really directed to the entire panel, and the question is that Africa has probably one of the greatest needs for low-cost proteins, and nobody's mentioned doing any plants in Africa. Does anybody have any plans to expand into Africa where they could really use a lot of help in both the waste treatment and production of protein?
I'll jump on that only because we actually have been working on one since March of last year. And it speaks also to the circular economy question, because you have fish processors on the west coast of Africa, Morocco, Mauritania, and further south, who have huge amounts of wastewater that go back into the ocean and pollute the bays. So for us, we can go into those sites, capture all of that protein, convert it to a single cell protein, but then create that water to recycle back. And if you think about it, especially Morocco and Mauritania, heavy desert areas, water is expensive. So we can bring the water back to the community for industrial purposes. We're not going after potable. But on top of that, we can then take the single cell protein, and if we don't take it out into the world into a feed ingredient, we can literally put it into the desert, and we've ran our own tests on this, and convert desert to farmland. And you actually make soil. You don't just provide a fertilizer, but the single cell protein goes in and creates a soil. You've also got your fresh water from the fish, because you can desalinate the fresh water out of the fish. And so you really have a major circular economy, which is especially valuable to the desert climates along the oceans. You know, it can work anywhere, but you don't have many large volume water producers inland in Africa. So you need to be on the coast in the desert areas for that.
Yeah, makes perfect sense. Thank you, Mark. Anybody else?
Yeah, I would just note that sea freight is an extremely efficient way to move large volumes of material intercontinentally or regionally. And when we're talking, most of the products under discussion in this call are moderately high value. And so Africa does not have to necessarily have local production to get the benefits of these products, when you can do the substantial value add of turning these protein meals into high value aquaculture species. And that's probably economically more important. In many cases, the question is, in the future, where is the natural gas? Where is the biogas? Where is cheap renewable energy? And there will be some answers in Africa, but there are going to be some areas which just don't have those resources. And so some of these, or where you can't make barley, right? You can't grow it effectively. So it's going to be on a case-by-case basis, I think.
Okay.
Can I make a quick comment?
Please.
I was just looking at the bioethanol industry in Africa, and it's not strong. But when we start turning what was traditionally a low-value co-product into a high-protein material, then that gives a whole new impetus to this biorefinery proposition. And so turning crops, to me, the vegetable crops are the base of the pyramid on which we build any production system in livestock production or food production. And if we can turn those crops into more valuable products, not co-products, but valuable products, then it gives impetus to the biorefinery concept. So I could see this sort of development picking up pace in countries like Africa, where we can produce the basic crop, but the fractionation of the crop into those components that we can use in livestock production is the key to the future.
Good, thank you. Okay, we've got a couple more questions, and one of them for Larry, for NIP Bio. feed kind. What can you explain how? Excuse me. Can you explain how the single cell proteins really are applicable to recirculating aquaculture systems?
Yeah, we actually see several opportunities within the RAS facilities. So potentially going in as a finishing feed, also as a juvenile feed, and really tailoring our platform, which is really what we are, to these different specific stresses. And water quality is just a really important, highly sensitive topic. You know, in these land-based systems, it's not like you have the tides that are washing away nutrients and dealing with it. So what I showed a little bit in terms of nitrogen conversion, but also there is a very important impact around off flavors as well, that we have some technology that we've developed. So yeah, there really are several different slices there for us to go after. That also, you know, the world of RAS is becoming more and more of a hybrid, as many hatcheries, of course, are land-based and ultimately maybe go into net pens. But, you know, there's still similar transition and stressful issues all throughout that growth cycle there.
Maybe I can add, Paul here, maybe can add that,
yes, please.
You know, RAS systems generally are very highly intensive systems. And that's a trend going in the industry to move to more intensive culture. And one of the important points there is that you need highly digestible, highly concentrated nutrients, because you don't want to create a lot of waste. So that's certainly an interesting driver for single cell proteins.
Okay, thank you. Yeah, that's good. Okay, I've got, I think we got time for maybe one more question directed to Alan with Calista. You said you're producing 20,000 tons of single cell protein. What do you think about marketing and competitive price of your product compared to fish meal? And how can you convince feed companies to use yours as their preferred protein source?
Yeah, I think, I mean, that's a good question. It's a kind of a question for everybody, right? So as far as marketing the product, we've had, you know, a lot of success already in China. We've got a technical team in China. We've partnered with Adissio, who has distribution throughout the entire region. They have a technical team that's traditionally been focused in poultry and livestock, but is really coming up to speed in aquaculture quite rapidly. And between the two of us, I think we're doing a very effective job of engaging the largest feed companies out there today. One, I think we're competitive on a value basis with fish meal, especially high-grade fish meal, especially in Asia where they're importing it from overseas. And then the second thing I'd say is that, you know, our work has shown that you can formulate on a no increased cost feed basis by really balancing nutrients. I mean, if you have a amino acid profile that's favorable, you can find other protein ingredients that match it and really support giving the same quality feed, the same nutritional value with a different basket than you started with at no increase in cost.
Okay, thank you very much. So before we wrap up, first of all, I just want to mention that we have a global audience of a little over 21,000 people today for the webinar. So I want to thank everybody for participating. And before we wrap up, I'd like to just give each of the panelists a minute or two for any closing remarks or announcements that you may have before we wrap things up. So Alan, as long as you're still there, why don't you, any last things you'd like to offer?
No, I mean, I would just say, you know, thank you for everyone who's tuned in. I think a big takeaway for me is, you know, not only is Calista at the brink of commercialization, but I think most of the other people are here too. And so it really bodes well for the industry and for aquaculture generally that so many alternatives are coming online.
Great, thank you. Paul, do you want to?
Well,
I
joined Alan in what he said. We as well. We are ready. We have our first plant, which is running. So I mean, you know, we're eager to start developing that market, and it's a market with great potential. And I think all of the participants have a great future in this market. So I mean, we should, you know, we are looking forward, and yeah. It's a very interesting seminar. Yeah.
Appreciate that. Thank you. Peter.
The biorefinery concept and taking crops and turning them into valuable products rather than co-products is the future. And I think, you know, we've all talked about volume, we've talked about competitive pricing, and that is all about getting the maximum efficiency out of the crops that we're producing. And we believe with what we're now doing with the bioethanol biorefinery that we're moving into a brand new future as to what we can do. And with some of the carbon capture techniques that we're using, CO2 capture techniques, we're going to make that whole process carbon neutral. So I think that's the future.
Great, thank you. David, say. NOVONUTRIENTS.
Yeah, I'm just observing that, you know, the majority of the folks on the call are producing, you know, thousands of tons a year or more, and they're all doing it in partnership with larger, more established companies, whether it's Russian Finance, ethanol industry, Scholar, you know, Cargill, BP, Blue Star Addicio, large beer companies. And so, you know, I think that's something to think about, is that the technology companies are going to work in partnership with established corporates who have access to a lot more resources, and that's really going to be the secret to getting to these, you know, high efficiency scales that we all want to get to.
Great. Okay. Thank you. Larry, please.
Some really great comments. I continue to be inspired by all of you, my fellow panelists. You know, I think it's a really exciting time in general. One thing I encourage us on sort of the pre-competitive type of sentiments is adoption, and ultimately not so much about our customers, but more about our customers, all the way down to the salmon on the plate. You know, and we're seeing some of the adjacent sectors like Veramaris and working with Spreading, working with some of the supermarket chains, right, to really kind of capture and adopt the whole value, because the risk, as those folks have pointed out in the past, is disproportionately on our shoulders, secondarily on the feed manufacturers, but the benefit accrues to all. And so we really, as innovators and entrepreneurs, need to disperse that more evenly. And it's ultimately the person who's eating that piece of fish, you know, they assume sustainability, they assume fair practice, but you know, how they value it in terms of their wallet, and how we get compensated for really driving that change in that future, is a delicate balance. And so it is something that we all need to be cognizant about, to make sure that these products do get uptake in the proper way.
Okay, good. Thank you. Yeah, Mark, any last comment?
Yeah, I'm going to approach it maybe as a group here, that we're really all on the same team again. We're not competing with each other, because there's literally hundreds of millions of tons of ingredients that are needed. And then I'm going to tap over to something that's right now surely on everyone's mind in the last few days, and that's the Seaspiracy Netflix show. And where that went, okay, with a message that's maybe partially right, but then maybe done completely wrong. And I think we have to think about the production of single cell protein and how someone might twist what we're doing, and instead of make it a good thing, try and make it a bad. So as a group, maybe we need to form an industry group that we stick together and help the market understand what we're doing, so that they don't turn it into some, you know, Franken approach, as opposed to a wonderful leverage of what nature already does. And so that would be one comment I have for the group, that maybe we we think about getting together as panelists and forming some type of a single cell protein production organization.
Yeah, that's an excellent point. You never know when somebody's going to twist the story around like we saw there. So, okay, Mike, any last comments?
Yeah, no, thanks. I guess I agree with a lot of the comments here, but I'd also like to compliment the F3 committee and people because you've put a number of these webinars together to help help communicate, help provide the right information. There's a lot of opportunities here, a lot of options, and also that not only is the feed industry looking for alternatives, but the industry in general is trying to provide those alternatives. And in those alternatives, we're also trying to become better stewards of our environment, of the earth, so that we can continue to, as mankind, humankind, continue to survive and thrive and grow and be responsible. So thanks a lot for what you're doing here and all the information that you've provided to the industry in whole.
Well, thank you. We appreciate that. And again, I want to thank all of you, the panelists here, for some great presentations and answers to questions. And again, I want to mention we had over 21,000 people on the live feed. We also have recorded this and it will be on a YouTube channel. So if anybody wants to share that, we're happy to share the YouTube address for other people to watch. And we believe that you guys, the single cell novel proteins are really taking the aquafeed industry by storm. It's really going to be interesting to see how things develop in the coming years. A lot of innovative tools, converting pre-existing waste streams and byproducts into feedstock. It's really going to be important, and especially with these nutritionally dense ingredients that you've got for fish and other livestock. And very important as we look at the supply of nutrients, and aquafeed companies are engaging in disaster preparedness, trying to de-risk their supply chain to stay ahead of shortages. It's all going to be really important. So we hope this seminar has been helpful, and please join us for the next webinar on algae and oil seeds in aquafeeds, which will be May 20th at 5 a.m. Greenwich Mean Time and 1 p.m. Beijing time. And also, before you go, please take a couple of minutes to watch our closing video here. And so we will run that, and thank you again for joining us.